Welcome to this video on the cell cycle, its mechanics, regulation, and pathology. We will explore the blueprint of cellular division, molecular checkpoints, and pharmacological interventions. The cell cycle, the engine of life, is a 24-hour circuit for human cells. It includes four main phases: G1, S, G2, and M. G1 is for growth, S for DNA synthesis, G2 for preparation, and M for division. Some non-dividing cells enter the quiescent G0 phase. Cell division varies between somatic and germ cells. Mitosis in somatic cells results in identical diploid clones for growth and repair. Meiosis in germ cells produces four unique haploid cells for sexual reproduction, introducing genetic diversity. Mitosis is the equational pipeline that includes prophase, metaphase, anaphase, and telophase/cytokinesis. It begins with chromatin condensing and ends with the physical division of the cytoplasm, yielding two identical daughter cells. Meiosis is the reductional sequence. Meiosis 1 separates homologous chromosomes, and meiosis 2, which is mechanically similar to mitosis, separates sister chromatids, resulting in four unique haploid cells. The cell cycle is regulated by rigid dependencies called checkpoints, which act as guardians. These checkpoints ensure flawless progression, checking for cell size, nutrition, DNA integrity, and spindle assembly before moving to the next phase. Guardian I controls size and nutritional status at the G1/S checkpoint. The cell monitors ribosomal mass as an indicator of nutritional status and uses geometric sensors in cylindrical cells to coordinate growth with cycle progression. Guardian II, the DNA damage response, is triggered by intrinsic or extrinsic factors. Sensors detect DNA lesions, activating processors like ATR and ATM. These activate brakes like CHK1 and P53, halting the cycle until damage is repaired. Guardian III focuses on replisome stability during the vulnerable S phase. Blockades can cause replication polymerases to stall. The mechanism stabilizes the stalled replisome to prevent lethal fork collapse until the blockade is cleared. Guardian IV is the spindle assembly checkpoint, ensuring accurate segregation of sister chromatids. It monitors kinetochore attachment and tension. If not properly aligned, a protein complex forms, preventing cell division and aneuploidy. Cyclin-dependent kinases, or CDKs, are the core masterminds of the cell cycle. They are inactive on their own, but become active when bound to cyclins. Checkpoints override this activity using inhibitory proteins like P21. System failure, often due to the loss of P53, abolishes the G1 checkpoint. This forces cells to rely on other pathways and can lead to uncoupling between DNA replication and mitosis, resulting in aneuploidy and mitotic catastrophe. CDKs are crucial therapeutic targets in cancer. Since P53 mutant cancer cells depend on G2/M checkpoints, highly selective CDK inhibitors like palbociclib can be deployed to halt proliferation by targeting the ATP binding site in the kinase pocket. The architecture of division is defined by conserved mechanics and stringent regulation. Interventions like CDK inhibitors can induce targeted apoptosis in cancer cells, restoring control and combating runaway proliferation.